Hello everybody, welcome to the second of our lectures for topics of biochemistry. In the first session we looked at the chemistry behind metabolism and the types of enzymes that carry out those biochemical reactions. And now we're going to turn to a little bit of an overview of metabolic flux and regulation, how metabolism or metabolic pathways flow through the cell and how they are regulated in various different ways to control that metabolic flow. So if you think about metabolism, it's basically a network of chemical reactions. Some precursors or substrates are used by many different types of pathway, and the end products of those pathways are maybe the starting substrates of others. And of course they're intertwined in such a way as to meet the needs of the cell, to meet both the energy needs of the cell and to provide it with the molecular building blocks to enable it to produce new molecules, to enable it to grow and divide. Now, flow through these metabolic pathways occurs via a number of different mechanisms, and of course they all talk to each other. So these are intertwined regulatory networks, and they can respond to changes by the cell itself, internal fluctuations, maybe substrate concentrations, the build-up of products, the requirements, the energy requirements of the cell, but also if any environmental conditions or external pressures are applied on the cell, they're able to respond to that and change metabolism appropriately. So if you like, here we have essentially almost the central dogma of biology, but explained in terms of regulatory networks. Down here we have what's called the open reading frames. So these could be the genes for a particular enzyme. These can then be transcribed and translated to produce the protein, the enzyme itself, which can then catalyze numerous different reactions, producing various different compounds, which can also then feed back and inhibit other parts of, so for example here alpha-D-glucose 6-phosphate can inhibit this particular isozyme of an enzyme to prevent it from producing glucose 6-phosphate or something. So we can have, the whole system can interact with each other, and they can go all the way back here and start to interact with the gene's transcription. That's how the level of control can be. So first thing though is, how big are we talking? What is the size of this network? How big is the metabolome? So back in 1955, not long after the structure of DNA was discovered, Donald Nicholson compiled all of the metabolic reactions known at that time. So this was 70 years ago, 65 years ago. And he, at that time, compiled 20 metabolic pathways. Roll on to the 1960s, just 10 years later, and it was thought that the majority of the major metabolic pathways had been defined. There had been an awful lot of work been carried out, and they were quite confident that by the 1960s the major metabolic pathways had been defined and worked out, and elucidation of the biochemical pathways was nearly complete. If you now compare that to the current days, well, the current view of is that metabolomics, which is the most up-to-date, high-throughput global experimentation technique that looks at all the metabolism in one go, has revealed this is not so. New metabolites and unexpected pathways and fluxes and within these pathways have been shown to have important physiological relevance and have been discovered. So actually it's a highly complex network of enzymes, of metabolites, all being controlled in different ways. Traditionally, we used to study one enzyme, one protein, and look at it in isolation. Nowadays, we know proteins and enzymes do not work in isolation, but they are part of these networks, and therefore we wish to study them on this scale. We wish to study them as part of these big networks. And so metabolomics, amongst a few other techniques as well, these other omic technologies, have enabled us to look at gene expression, to look at protein development, to look at enzyme activity in terms of metabolomics on a global scale, and manage to compare different situations to enable us to understand what's going on within the cell. So one of the things that we need to study is metabolic flux. And what do we mean by metabolic flux? Well, the basic requirement of any living system is that it needs to adapt its metabolic needs to changing environments. We're all, I'm sure, hopefully aware of the term homeostasis. So these adaptations to our new situation, either through internal changes or through external pressures, they can be quick and mild changes, or they can be very slow and quite large changes or drastic changes. So cells therefore have to constantly monitor their metabolic state in order to make sure that they're meeting the cell's needs, they're meeting its own needs. First thing we need to know is, well, how do they do this? And secondly, how can they regulate their metabolic pathways? Now, there has been renewed interest in metabolism because of possible intervention points to treat diseases. So having an idea of how metabolites are produced and how particular metabolic pathways, the flux through metabolic pathways, what's the critical steps in metabolic pathways, the rate limiting steps within metabolic pathways are quite important in terms of disease, because you may want to modify those in certain disease situations. So that's why there's been a renewed interest in identifying what we call regulatory nodes, the key regulatory points within the metabolic pathway, which can be useful to exploitation by new therapies. Now, this is of course challenging due to the very complex structure of all these intertwining metabolic networks. So, metabolic flux analysis, what this is looking at the rates or the in vivo velocities of metabolic reactions and looking to see which enzymes and which components are responsible for determining the rates of that particular reaction. In other words, which steps are known as the rate limiting steps. So, transformation of these intermediates by enzymes and the transport of metabolites between cellular components have been identified as particularly important in controlling and regulating metabolic pathways. This depends upon the systematic organisation and the interplay of the enzymes themselves within the metabolic pathways, the transport proteins or carriers responsible for shuttling intermediates around the cell and across membranes, and also the concentrations of substrates within the cell or may be available within the environment outside of the cell. Now, all of this knowledge is essential to help us to work out how a particular metabolic pathway is regulated, and also to augment our understanding of how metabolism is embedded in cellular decisions. How does the cell decide to upregulate this particular pathway, downregulate this particular pathway, for example? And this is very important, of course, within disease cells, because information about the flux within key metabolic pathways will help us to identify and select particular therapeutic targets, particular enzymes within those metabolic pathways that we wish to target, perhaps with substrate analogues or product analogues that can regulate enzyme expression, enzyme activity. So, if we think then about a particular metabolic pathway, this goes back to when Buckner first started to dissect the fermentation pathway within yeast, there was the conventional wisdom that the rate of a metabolic pathway was dependent upon the enzyme that had the slowest rate of catalysis, the slowest or activity. So here we can see I've got a metabolic pathway here going from A to B to C to D to E. So these are the, if you like, the substrate and its intermediates, with the product being E. So this is the substrate A, then we've got intermediate B, C, D, and E. And they're each step is catalyzed by separate enzymes, E1, E2, E3 and E4. Now, conventional wisdom, what used to happen in the past would we think that one of these enzymes, once we looked at the rate of the reaction, was responsible for how fast this metabolic reaction would go. So, for example, let's say it's E3. So let's say E3 was the slowest rate of the reaction. In other words, it didn't really matter what E1 and E2 and E4 were doing, because E3 was the slowest one. And if we could then somehow increase E3, the concentration E3, then maybe we can increase the rate of the reaction. So this is what happened. And this happened until really genetic engineering came along, and we could start playing around with the genes that control of each one of these enzymes, and we managed to upregulate we upregulate or expressed higher at higher concentrations enzyme 3. So this was genetic engineering then allowed us to test this hypothesis, and what we did was we increased E3, and what is predicted from our conventional wisdom was that the rate of the production of E would increase in proportion to the increase with E3. Did this happen? No. Most often no. In fact, we found it didn't happen, and that actually sometimes it did, but majority of the times it didn't. That actually by increasing the known rate limiting, the one enzyme that increased the rate limiting step, did not always increase the proportion of the product formed. So the control of the flux for the metabolic pathway, we found out, was actually not due to one enzyme, but actually distributed among several enzymes within the pathway. So this makes things a bit more complex. And in fact, it depends upon the metabolic circumstances that the pathway finds itself under. And these are the things we need to consider. We need to consider the supply of starting material A. Is there a ready supply? Are we dependent on it being transported into an organelle, for example? There may be a metabolic requirement need for other pathways. So here A is going to B to C to D to E, but actually maybe B is also an intermediate or a substrate for another pathway. There's a branch pathway and B comes off down here. So not all of B ends up going down this pathway, but maybe siphoned off into a separate metabolic pathway. The effects of each one of these metabolites might have regulatory roles. They may feed back and inhibit enzymes further previously within the metabolic pathway, thereby regulating its own production. And there may actually be external influences upon this pathway too, such as hormones, insulin or glucagon, for example, may actually affect the activity of particular enzymes. So all of these things can then affect the rate limiting step within a metabolic pathway. So determining the flux is a bit more complicated than just looking to see which enzyme is the slowest catalysis rate. However, we can experimentally determine the contribution of each enzyme to metabolic flux, and that's really useful if we want to know what type of, especially in a disease situation, we want to know which of the enzymes is what their contributions are. So as an example here, I'm going to turn back to our favourite metabolic pathway, glycolysis, and look at the first part of my glycolysis. And in this particular example, what I'm going to do is I'm going to homogenise rat liver to release the soluble glycolytic enzymes and use them to carry out the conversion of glucose to fructose 1-bisphosphate in an in vitro experiment. So just to remind you what the first steps of glycolysis is, here's glucose. And glucose is phosphorylated by hexokinase, so the phosphate group is transferred from ATP onto glucose to form glucose 6-phosphate. Then glucose 6-phosphate, what we want to do here is we've got the aldehyde here, we want to turn this into a ketone, so phosphoglucose isomerase removes the carbonyl group from the first carbon to the second carbon here, and then finally that gets phosphorylated again using another molecule of ATP, and the phosphate group is transferred onto the carbon here, okay, to make our fructose 1,6-bisphosphoglycerate. And what we're going to do is we're going to see out of these three enzymes which ones contribute the most to the overall rate of the reaction, which ones actually affect, the flux through here is dependent on which one of these enzymes. So if we do these experiments, we can see this graph. So this graph here has enzyme added, amount of enzyme added, so we're adding more enzyme along the bottom here of each one, and here we've got glycolytic flux, so that's the rate through this pathway from going from glucose to fructose 1,6-bisphosphate. And here we can see phosphohexose isomerase, here phosphofructokinase 1, and hexokinase. So hexokinase is the first enzyme in our metabolic pathway, and we can see that as we increase enzyme concentration, so the activity, the glycolytic flux, whoosh, shoots up. Okay, so it is proportional. So the more enzyme we add, the greater the flux. The more of this we add, the greater the flux. Okay. So increasing hexokinases increases the rate proportionally. If we look at phosphofructokinase here, which is the next one down, again we can see that as we increase the enzyme concentration, so we get an increased rate of flux, but maybe not as well, certainly not as much as we saw for hexokinase. Okay, so increasing phosphofructokinase also increases the rate too. However, if we increase the concentration of phosphoglucose isomerase, nothing happens. We do not increase the flux rate going from glucose to fructose 1,6-phosphate. So here, increasing phosphoglucose isomerase has no effect on the overall flux of turning glucose into fructose 1,6-bisphosphate. What this means then is that these enzymes, hexokinase and phosphofructokinase, these set the flux through the pathway. These are the enzymes that determine the rate that the pathway runs at. Obviously, hexokinase more than phosphofructokinase, but these are the enzymes that are actually important in controlling and regulating the flux through this pathway. As you can see, phosphoglucose isomerase has no effect at all. Okay, so this is really nice. This enables us, we can look at each one of these enzymes in between, and we can see the effects it's having on the overall flux through our pathway. And we can actually then do this, we get some quantitative information out of this that we can then use to model, if you like, our pathway. So the flux control coefficient, which has been given this symbol capital C, it allows us to quantify the effect of the change in enzyme activity on metabolic flux through our pathway. And it expresses the relative contribution of each enzyme to setting the rate at which metabolites through a pathway. Okay, so the coefficient is not a constant because it's a coefficient, which means it will change depending on metabolic circumstances. But what it does do is it gives you a measure of the contribution of that particular enzyme to the overall rate within that pathway. Now, flux is given by the overall flux, if you like, is given by given by the letter J, capital J. Now, if the flux control coefficient C is 0, then this enzyme has no influence or impact on the flux. Just as we saw for phosphoglucose isomerase, that did not do anything. We increased the concentration, so it has a coefficient of 0. If the coefficient is 1, which is the other end, the other extreme, this means that the enzyme wholly determines the flux of the pathway. That's basically our old wisdom of that whatever the rate determining step is dependent on the slowest enzyme. That essentially one enzyme. That's the other extreme, essentially, that one enzyme is responsible for the entire rate within the pathway and everything else doesn't have any contribution at all. Of course, in reality, most things are somewhere in between this. The other thing you can also have is a negative flux control coefficient, and that's because if you've got a branched pathway, here you have an enzyme that draws intermediates away from the other branch, and this of course can have a negative influx on the flux of the other branch. So here we've got our pathway going A, B, C and D, but here you can see that B is siphoned off in a branch pathway to produce E. And enzyme 4 here, has a negative coefficient with respect to this pathway because it's removing flux from this main pathway. So you can see that all of A does not go to D. Some of A gets turned into B, but then some of B gets siphoned off to E, and the rest goes on to D. So again, the contribution that enzyme does by the activity of that enzyme is given by this negative coefficient. Okay, so this is a negative flux control coefficient with respect to this pathway because that intermediate B has been siphoned off. Okay. Now, if we look again back at our original example of glucose and add some numbers to this, we can see that if we put the coefficient for hexokinase here is 0.79, for phosphoglucose isomerase is exactly as we predicted, is 0, it has no effect, and of course for phosphofructokinase it's 0.21. Now I hope you've noticed this already, but if you sum all the coefficients, then the pathway, if you sum all the flux coefficients in a pathway, they've got to equal unity or got to equal 1. And if we go back to the previous one here, we can see that again this still holds true. Here's our pathway A going to D. All these coefficients have to add up to 1. Okay, and then we can see that here 0.3 plus 0 is 0.3 plus 9 is 1.2. But because the 4 is involved in here, we've got the branch pathway. This is a negative coefficient number. We can see that 1.2 then becomes 1 because we're siphoning off 0.2 in this branched pathway here. So the important thing is that all the coefficients of a particular pathway must equal unity or must equal 1. And so therefore these coefficients therefore do give you a proportion of the contribution that that enzyme is having towards the overall pathway, the overall flux through that pathway. And here you can see quite clearly hexokinase is having the greatest contribution towards that to determining the flux. The next coefficient is the elasticity coefficient, and that's given by this eta here. And it expresses the responsiveness of a single enzyme to changes in the concentration of a metabolite or regulator. So this tells you a little bit more about a particular enzyme and how it's sensitive to changes in concentration. And again, it's based upon the enzyme's intrinsic kinetic properties. So it's based upon V and Km. So if you look here at low concentrations on our standard rate curve here, we've got substrate concentrations increasing along the bottom and velocity up the side here for a particular enzyme. At low concentrations, say well below the Km, if we increase the substrate concentration, we see a comparable increase in the rate. Yep. So as we increase substrate concentration, so the rate's going up. That means that the elasticity is near the extreme here, 1. Yep. You change the substrate concentration, rate changes. Now, if you look at really high concentrations, where you're way, say, 10 times above the Km, up here, say, then if you increase the substrate concentration, you're not seeing any difference in rate, because what's happening here, of course, is that the substrate here is well in excess of the enzyme concentration, and the enzyme's going flat out. It's approaching Vmax. So no matter how much you can increase the substrate concentration, you're not going to increase the velocity. Down here, of course, where the substrate concentration is much lower than the enzyme concentration, increasing substrate concentration results in a corresponding increase in velocity. So up here, where you increase the substrate concentration and you're not seeing any further increase in rate, this means the elasticity is 0. Yep, no matter how high how more substrate you can put in, you're not going to see an increase in rate. The final of our coefficients is the R coefficient, or the response coefficient, and this represents the quantitative impact of the influence of an external factor on the influx on the flux of our pathway. So this is like adding a hormone or growth factor to our system and then seeing what it does to our enzyme. It's not a metabolite and it's not another enzyme on the pathway, it's purely an external influencer that comes in and affects how our pathway runs. Now, experimentally, how you do this is you look at the response of the metabolic pathway measured at various concentrations of the influencer to obtain the response coefficient. So you can work out, at different concentrations, what the response, how each one of our metabolic pathways responds to that. Now we can relate all these coefficients to get a measure of the responsiveness of a pathway to an outside factor that affects a certain pathway. It's a function of the three things. It's a function of how sensitive the pathway is to changes in enzyme activity, i.e. the flux coefficient, and also how sensitive the specific enzyme is to changes in the outside controlling factor, and that's our elasticity. So here we've got our response coefficient is equal to our flux coefficient times by our elasticity coefficient. And what we can do is we can look at every single enzyme in our pathway separately and by determining each one of these factors for each of the enzymes and putting them together, it allows us to predict how the flux will change with respect to changes in any of the controlling factors of the pathway. So if we say increase the substrate concentration of an intermediate, if we increase the concentration of an external influencer, then we can predict what's going to happen with the overall flux through that pathway. So this is quite important, especially when looking at enzymatic pathways in drug discovery, to find out how enzymes are going to behave, or how substrates maybe react, how substrates are converted into particular products, and how what is the control of that particular pathway to enable, so you can work out then which would be the main targets to attack, or to design your new therapy or new therapeutic drug against. So if that's flux, what about regulation itself? How do we regulate metabolic pathways? How is regulation of this complex, massive metabolic network carried out? So again, if we define what control and regulation are, so if control is where we have regulated enzymes that themselves control the flux through the pathway, where if we can think about regulation as being rebalancing at the level of the metabolites in response to a change in the flux. So so the cells will if there is a change in the flux, then the cell will try to rebalance those levels of metabolites, and that's occurred by regulation by various methods to reach homeostatic levels of metabolites as before. Now, how we can do regulation are lots of different ways. If we're looking at, we can start off by the gene level. Yep. So we can encode different forms of the same enzyme which have slightly different substrate specificities or work at different rates. So these are isoforms or isoenzymes. So they carry out the same metabolic reaction, but they may be controlled in different ways, i.e. responsive to different regulators, external influences, and also may have slightly different specificities. We can work at the transcription level. We can activate genes under certain situations or turn them off. Yep. So if we've got a particularly high concentration of metabolite, we might want to turn on the enzyme that acts on that metabolite. Yep. So the gene gets expressed, protein gets made, more protein, faster rate. Conversely, too much, turn off the gene expression, protein translation goes down, less of the protein available, enzyme activity flux decreases. We can have alternate splicing. Again, this is making different forms of an enzyme. Yep. So in higher eukaryotes, we have coding and non-coding regions. We can then select which of those regions come together to make particular forms of a protein. There's messenger RNA stability. We can control the time that a messenger RNA is translated. So the shorter the time a messenger RNA is around for, the less protein is made. Less protein, less enzyme, less activity, etc. So you can turn things on and off quickly. And again, we can regulate translation, the effects of translation, and how fast translation occurs. Or in fact, we can just degrade the protein and how proteins degraded. Are the proteins turned over, the enzyme we're making, is it turned over very quickly, or is it turned over more slowly? Now, these are all what's known as course level regulation. Changing levels of expression. It's all quite on a large scale. Now, and these also take quite a long time. These can take hours to days to produce. So we're looking long-term changes to enzyme activity, to flux activity here. And so, because they're really all things that control protein abundance. However, we also can have control at the level of the local level, in saying local concentrations of metabolites. Remember, substrate concentration is very is key to affecting perhaps enzyme activity. Allosteric regulation of enzyme activity, where things bind to enzymes and change their activity. And again, post-translational modification of enzymes, such as phosphorylation, dephosphorylation, turning on and off enzymes, activating them, deactivating them, etc. Now, these are all fine level changes in regulation, and they can happen in seconds to minutes. So this is really at the level of the current demands in the cell. Then it's these sorts of things that are responding to the changes that are happening inside the cell. So if we have a look at metabolic regulation overview, this is quite a nice diagram because it shows you both the coarse level and the fine level control. This on this side really is coarse level. Yeah, we're having extracellular signals are saying turn on. We need more protein. This then turns on transcription factors, which turn on genes, gene expression, protein production. Here we are, translation, messenger RNA degradation to get rid of signal. We've got protein turnover, where the protein may be created and then destroyed. But eventually we end up with this thing called enzyme abundance. So this is how much at the end of all of this we have a certain level of protein that can carry out the metabolic reaction. That gives us, if you like, our enzyme concentration at a homeostatic level. We can, of course, make more by turning on transcription. we can, or make less by turning off transcription, or we can control the rate to which the protein is degraded. But eventually we'll get to a sort of steady state level of enzyme abundance for a particular metabolic situation or environmental condition. If we then look at metabolic flux, which is the thing we're trying to measure, the other thing that can affect our metabolism, our metabolic flux, is the effects of cofactors. Remember, enzymes just don't work alone. Enzymes also have metabolic teeth, cofactors, coenzyme, and again we may be able to control those. We may have high levels of NADPH or low levels of ATP. These may signal particular metabolic conditions inside the cell, which therefore may mean turning on enzymes, turning off enzymes, or may actually just limit the rates of reactions that can actually occur by not having a lot of these around. You may need NADPH or NAD for a particular reaction. If there's not much of it around, again the reaction is going to be slow. You may need ATP. Again, if there's not much ATP around, the reaction is going to be slow. Okay. Another thing is compartmentalization. Where do these reactions occur? Compartmentalization may be that you've got reactions going on in mitochondria inside endoplasmic reticulum, which is great for making specialization cells, but you may need to get the substrates and the cofactors into these compartments, which means they may have to cross semi-permeable membranes, which means that their rate of entry or their rate of availability depends upon how quickly they can be transported into that particular organelle. Again, so there's another form of regulation there. So these, and then we've got the fine-tuning regulation here, which is one we just talked about, where we have allosteric regulation, where substrate goes to product, and we have an allosteric inhibitor here, which binds at a site separate from the active site, changing and modulating that enzyme's activity. We have substrate availability. Local concentrations of substrate may mean that the will give you the rate to equilibrium. It will determine how quickly you can turn the substrate into product. There's low concentration of substrate, the enzyme activity. You're going to get much product formed, for example. If you have high concentration of product, you may actually go the other way, and you may end up forming whatever, like substrate. There may be covalent modifications. This is like phosphorylation. Here we've got ATP going to ADP, and we've got phosphorylation of our enzyme to make an activated or inactivated form of that enzyme. And again, we may actually have the binding of our protein, our enzyme, to a regulatory protein, which itself is controlled by some other cell signaling system. So again, which may turn on and turn off the activity of our enzyme, and therefore regulate levels of substrate and product within inside our cells. So we're going to have a little bit more a look at these in each of these different types of metabolic, of these different regulatory points, and maybe some examples as well.